So I met Eric much later. He showed up at the museum, the Smithsonian's National Museum of Natural History, in the late 1990s with Mike Levine and Alan. And he was on a mission, he and Mike, to convince the administrator of NASA that NASA should support evolutionary biology. Now, in the best Eric fashion, Eric actually didn't mean evolutionary biology, because that would include things like population genetics, which, as far as Eric was concerned, was, well, I won't describe Eric's views in population genetics, but he had a very specific goal in mind for NASA, which was to support work on the evolution of gene regulatory networks, which was, as far as Eric was concerned, about all there was that was interesting in evolution. As part of that trip, he wanted to see the fossils of the Bruges Shale, of which I'm the curator at the Smithsonian, and his postdoc Kevin Peterson, who I knew, had said, "Well, you should give this guy Doug Erwin a call, and he'll show you some fossils." So Eric and I arranged this visit to the museum. What Eric didn't know was that when I was an undergraduate, I had devoured the 68 and 71 and 73 papers and virtually memorized them. And so when, as I was showing him the Bruges Shale, we started talking about the gene regulatory network papers that he had done then, and also the work that had been developing in the late 90s in the field that we now call evo-devo. As a paleontologist, I had been very concerned, along with two of my colleagues, Dave Jablonski and Jim Valentine, about the inferences that many developmental biologists were making about the nature of the last common biotinian ancestor. It mattered a lot to us as paleontologists because, in a sense, the holy grail of both evo-devo at that time and of paleontology was trying to understand the nature of the last common biotinian ancestor, the ancestor of flies and chickens or mice or whatever your favorite bird is. The inferences that the discovery, just to recall to you the situation in the late 1990s, people had discovered an incredible, including David and others, had discovered the incredible deep conservation of developmental genes across biotinians. It wasn't yet clear the extent to which many of these genes were also found in other forms of metazoans like Mendelians and fungus and trichoplax and things like that. That would come later in the 2000s. But the papers that were being published in the late 1990s had suggested that this last common biotinian ancestor was morphologically and developmentally quite sophisticated. It had eyes, it had a heart, it had a brain, it had a tripartite brain, it had a whole lot of other morphological features. And during this visit to the museum, while I was showing Eric the fossils of the Bruges Shale, we got to talking about a lot of this developmental work, and it turned out that neither Eric nor I believed, well, we believed some of the published work. We didn't believe a lot of the rest of it, and we thought that it had been horribly overinterpreted. Much to my surprise, after I got Eric to sign my undergraduate Xerox copy of his 1968 paper, Eric invited me out to Caltech. I'm from California, so I got to Los Angeles a lot to visit my parents. And I went by Caltech and spent a couple of days there talking to Eric, and out of that eventually came our first paper in 2002, which was a reinterpretation of much of the developmental data that had been produced by evo-devo studies over the previous 5 years or so. But I met Eric at what turned out to be an enormously fortuitous time, because the lab, as Ellen has just described, was in the midst of this transition, and they had begun working on the Endo-16 system, which opened a doorway into really working out the complete regulatory genome of Strongylosis introitus up to 30 hours, which was Eric's goal at that time. So there was, every time I talked to Eric on the phone or went out to Caltech, there would be a whole set of new discoveries from the lab and new things for us to talk about. Although our first paper was really on this issue of what the last common biotonic ancestor might have looked like morphologically and how it might have operated developmentally. After that paper came out, we sort of transitioned into an interest in how gene regulatory networks evolved over time. And that came about because one of Eric's deep interests, the reason he came out to see the Bridge of Shale in the first place, was the nature of development and its role in constructing body plans, particularly during the Ediacaran and Cambrian radiation. And which is also one of my great interests. So out of our shared interest in the origin of body plans, Eric and I began a collaboration. Each paper ended with my telling my girlfriend at the time and now wife that was the last time I was ever going to do anything with Eric. Because I couldn't take it anymore. He was an amazingly insightful scientist. He had incredible an incredible breadth of knowledge and incredible powers of logic in order not only to understand the nature of development and gene regulatory networks, but also a host of other fields. When I began this collaboration with Eric, I didn't know that it was going to be a collaboration that would last till the end of his life. I didn't understand that my role was not simply to provide develop geological and paleontological context for these discussions. I quickly realized that he often read the geological papers and the supplemental information in as much detail as I had. We would wind up in lengthy discussions about one particularly notable time from one of the 2009 papers. We spent months arguing over the sequence stratigraphy of rocks in southern China between the Yangtze gorges and the southern areas around where the Doshanto embryos, these fossil embryos, were found. I sort of figured that as a paleontologist at the U.S. National Museum with a PhD in geology and 15 years worth of field experience in the geology of China, I had some minor credibility in this area relative to a developmental molecular biologist who had spent about 2 days doing field work in China. I was quickly dissuaded of that opinion as we began this detailed argument over how to correlate rocks between the Yangtze gorges in southern China, which lasted over 6 months or so of writing one of the papers in 2009, 2010. Part of this came about because there were varying estimates for the age of these fossil embryos that Eric had a side project that I'll describe in a moment. And Eric, at some level, very much wanted them to be the oldest evidence of animal life. There were aged Lutetian Hafnian ages to suggest that these embryos could be about 600 million years old. I thought the better uranium lead date suggested an age of about 580, and we were attempting to construct a very simple figure that should have taken maybe a half an hour, tops. It took close to 6 months. At one point I suggested that we could compromise and say that EHD believes the age of the Doshanto embryos, there's laughter down here because the compromise was not really in Eric's vocabulary. So there was a huge explosion over the suggestion of a compromise. And eventually we worked through this and came up with a figure that both of us could agree on. I was absolutely sure that I was right. Eric was absolutely sure that he was right, given, of course, his vast knowledge of Chinese stratigraphy. The denouement of this came last January, when a year ago, July, when I was in China for about 3 weeks looking again at these rocks. And I discovered that there were new uranium lead dates that showed that Eric actually had been right all along. That's the sort of thing that you had to deal with Eric, was even when he wasn't right, he somehow managed to worm his way out of it, or his intuition gave him some insight. Now, the fact of the matter was that when we were doing this in 2009, neither one of us really had enough reliable geochronology to know quite what the age of these rocks were. But it was that the give and take and the battle over the details of the geology of South China that made working with Eric both invigorating because of the depth of understanding he had not only of development but of evolution in other fields, but also the detail that he demanded of you, and frustrating because of the 6 months that took us to resolve one figure in the paper. In that paper, actually, because of the interactions with the editor, I wound up banning him from any correspondence with the editor and told him that I would take care of all the conversations with the editor, because if Eric had kept up conversations with the editor, we never would have gotten the paper published. Let me close with two other comments. I've recently been rereading the Isaiah Berlin essay on the fox and the hedgehog, which has led me to think a little bit about Eric in this sense. You'll recall that Isaiah Berlin originally wrote this essay in the 1930s, and it was based apparently on a game that often was held at Oxford colleges, thinking about whether somebody was a fox or a hedgehog. Fox, I remember, knew many things, and the hedgehog knew one great thing. And Berlin, in this essay, which is really about Tolstoy's view of history and the appendix to War and Peace, argued that you could place many artists and writers and other figures from history in either the category of a fox or a hedgehog. Marx was a fox, was a hedgehog. Many other people were foxes. And Eric, I think, clearly belongs in his scientific career within the field of the hedgehogs. The people that know one great thing that provides this overarching view of how life works. And for Eric, that was the importance of gene regulatory networks for cis regulation early in his career, and then genomic regulatory structures and networks toward the end of his career. But the point, one of the other points that Isaiah Berlin makes in this essay is that nobody is wholly either a hedgehog or a fox. And that was, of course, true of Eric as well. Eric had, as we'll hear this morning from others, and as Erwin has just said, he had all these other sides to him. When I went to Naples to get a talk at the Marine station there, I discovered that Eric loved visiting Naples, not just because of the food and not just because of the scientific colleagues there, but also because he could indulge his interest in Roman history, of which he had a very deep knowledge. So there were all of these size to a very complicated man. The part that I was privileged to know best was that was very interested in the fundamental problems of how we understand the origin of body plans and one of the most fundamental evolutionary transitions in the history of life, the episode of the Cambrian. And it might seem paradoxical that somebody who's primarily experimentally interested in modern sea urchins was able to project back 600, 550 million years and apply the experimental work on modern organisms to events 600 million years ago. But I think that was really the power of Eric's intellect, was his ability to do that. Thank you.